Hi everyone, I'm Isidora and I'm a PhD candidate at the University of British Columbia, Canada and AFC Canada. And I first want to thank Banff Vintners and the American Wine Society Educational Foundation for supporting my studies about predicting new threats to the North American wine and grape industry. The changes in climate patterns and the plant movement around the globe has helped fungi to overcome geographical barriers. So pathogens that were once limited to one area due to dispersal or climate limitations can now have new opportunities of new interactions with new plants. And the problem is that we don't know what the outcome of those new interactions will be. Maybe those grapevine pathogen interactions will turn out to have very minimal symptoms, causing no problems to the industry. Or we might be witnessing the rise of a new outbreak. Very extreme possibilities, but the problem is that if we can't predict them, we can't prevent them. So my thesis focuses first on how can we predict new fungal grapevine associations. And for this, I use the Butysphoraceae fungal family, known to cause the Butysphoria dieback, one of the worst grapevine trunk diseases. But predicting a new jump of a pathogen to grapevines does not tell us anything about how severe that new interaction, that new disease will be. So in the second part of my PhD, I focused on how can we predict the virulence of these new pathogens. So I used models combined with experimental inoculations of fungi into grapevine canes to see if we can predict how severe those new threats can be. So we can pay more attention to those pathogens that can cause the highest impact to the grapevine industry. So the idea is to predict, to prevent pathogen emergence in grapevines so we can keep our vineyards healthy.
Hello, my name is Douglas Vines. I'm a master's student in the Grape Breeding and Enology program at the University of Minnesota, and I am studying the phenotyping and genotyping of cold hardiness in hybrid grapevine. One of my projects utilizes differential thermal analysis, or DTA, to estimate the cold hardiness of grapevine buds at specific intervals throughout the winter. This allows us to better understand the patterns of cold hardiness in our breeding program and will aid us in the selection of breeding parents in elite progeny. My other project aims to identify quantitative trait loci, or QTL, associated with day length sensitivity in fall acclimation. We would like to select for the ability to acclimate in the fall based off of the day length rather than the temperature, as we believe that it will allow for more stability in acclimation as our climate shifts. I am utilizing shade cloth to control day length and am assessing acclimation response in 4 seedling populations. As our climate changes and our winters become more variable year to year, it is important that we are developing new grape cultivars that can withstand a variety of winter conditions. My work is supporting these breeding efforts. I would like to thank the American Wine Society for their generosity with the AWS Education Foundation Scholarship, as well as Virginia Weidler for sponsoring this fund. As a graduate student, these funds are so important in supporting our ability to continue pursuing cutting-edge research. Thank you.
Hi, my name is Arpa Boghozian, and I'm a fourth-year PhD candidate in the Horticulture and Agronomy graduate group at UC Davis. My dissertation project is studying the effects of grapevine red blotch virus, also known as GRBV, and its effects on grape skin cell wall metabolism and pathogens-related proteins, particularly in relation to phenolic extractability. It disrupts the ripening process, resulting in wines with lower sugar content, reduced anthocyanin levels, and higher acidity. These compositional changes in the grapes translate to wines with less color, diminished fruit aroma, and weaker mouthfeel. Economically, the virus can cost growers anywhere from $2,200 to $68,000 per hectare. My research focuses on 4 main objectives to better understand the biochemical effects of grapes throughout ripening. I collected grape samples for 2 years at 4 key stages of ripening: pre-veraison, veraison, post-veraison, and harvest from a commercial vineyard in Paso Robles, California. Specifically, I examined the changes in grape skin cell wall composition, the activity of enzymes involved in cell wall softening and ripening, and the expression of pathogens-related proteins in these grapes. All of these factors are then linked to the extractability of phenolic compounds in both grapes and the wines produced from them. Ultimately, I hope that this research provides deeper insights into how plant-virus interactions affect grape development. By understanding these biochemical disruptions, we can work toward more targeted solutions for both grape growers and winemakers. I would like to sincerely thank the American Wine and Education Foundation and the AWS chapter of Cleveland, Ohio, for their generous support of this research.
My name is Hannah Frank, and I am a PhD candidate at Cornell University studying under Dr. Justine Van den Heuvel. My research focuses on arbuscular mycorrhizal fungi, which are beneficial soil fungi in the vineyards here in the Finger. The goal of my research is to understand what soil, environmental, and management factors influence these fungi to help the growers here in the Finger Lake and across North America. I'm conducting research on 10 vineyards across the region. Every year at bloom and after harvest, I take soil and root samples from all of my vines. I then send the soil samples off for nutrient analysis. The root samples are split and half are cleared and stained, and then I assess them under the microscope for fungal structures to assess the colonization rate. The rest of the root samples are stored and will be used for DNA extractions to identify the fungal species that are present. Because of the increasing interest in these organisms, every year at bloom, I inoculate half of the vines at each location with a commercial inoculant. This is so that I can assess any potential impact on the colonization rate of the roots with this addition of an outside inoculant, but also so that I can assess any potential benefit to the vines of adding these extra mycorrhizal fungi so that I can give growers an educated assessment of whether or not it would be worth them investing in these commercial inoculants. I would like to sincerely thank the Meadowlands, New Jersey chapter of the American Wine Society and the entire American Wine Society Education Foundation for their support of my work. Thank you so much.
Hello, my name is Jesse Stevens. I would first like to thank the American Wine Society Education Foundation for their gracious scholarship. I'm currently a student pursuing a master's degree in viticulture at Washington State University. For the research I'm involved in, the overarching goal is to help growers minimize the risk of fungicide resistance in the pest grape powdery mildew by utilizing alternative fungicides or by implementing better use of current fungicide techniques. One of my projects is seeking to utilize UVC light as a means of managing powdery mildew in vineyards. UVC light produced by specially designed lamps can be applied onto a grape's canopy. Much like how we can get sunburns, the high-energy UVC light can cause damage to mildew and even kill colonies. A part of this project also includes looking at how UVC applications are affected by different canopy management techniques, such as removing leaves and removing shoots. The hope is this with research, we can provide an alternative pesticide that can be safer for growers to use and reduce risk of fungicide resistance. My other project is doing some myth busting around the use of sulfur in vineyards. Sulfur is an effective fungicide and powdery mildew will not develop fungicide resistance to it. However, during certain times of the year, growers do not use sulfur due to fears of it causing chemical-related burn injury on the plants called phytotoxicity, specifically during high-temperature parts of the year. The cause of sulfur burn on Vitis vinifera grapes, particularly in a dry western climate, have not been tested. Therefore, my goal is to determine what factors or combination of factors, such as temperature, humidity, and sulfur formulation, and what thresholds need to be met in order to actually cause sulfur burn. The goal of this project is to give growers a scientifically informed understanding of when they should and should not use sulfur in the vineyard. This will allow for more use of sulfur and decrease the risk of powdery mildew developing fungicide resistance. Thank you.
Howdy, my name is Michael Cook, a viticulturist with the Texas A&M AgriLife Extension Service. I'm also a PhD student in the Department of Horticultural Sciences at Texas A&M University. It is an absolute privilege and honor to be a recipient of the AWSEF Scholarship, in particular the W.J. Rochelle Endowment. I want to talk a little bit about my passion for not only Texas wine, but American wine in our industry, as well as a project or my project that I'm conducting across the state of Texas with the goal of providing research-based information and so really solutions for our Texas growers for long-term sustainability. I've been in the industry for over 10 years, and I've had the privilege of building strong relationships with our industry and listening to them and seeing what their needs are to these challenges. We all are facing challenges in the American wine industry, both on the growing side, the winemaking side, and even the selling side, the business side of things, through the tasting rooms, etc. One thing, though, on the viticulture side that we're all concerned about, no matter where you are, in most cases, is water scarcity. And also, how can we do more with less? And so in Texas, I established the most comprehensive irrigation and grapevine water use efficiency study. To date, this is our third season. We're generating a lot of good data that we'll be able to comb through and provide our growers with research-based information on not only the best ways to schedule and monitor irrigation and also water in the soil and vines, utilizing a lot of affordable technologies that can help improve our decision making. So we're excited to be able to continue with this work, to be able to get this information in the hands of our growers that they can add to their vineyard management protocols as it relates to irrigation and with a focus on water conservation. Howdy, my name is Michael Cook, a viticulturist with the Texas A&M AgriLife Extension Service. I'm also a PhD student in the Department of Horticultural Sciences at Texas A&M University. It is an absolute privilege and honor to be a recipient of the AWSEF Scholarship, in particular the W.J. Rochelle Endowment. I want to talk a little bit about my passion for not only Texas wine, but American wine in our industry, as well as a project or my project that I'm conducting across the state of Texas with the goal of providing research-based information and so really solutions for our Texas growers for long-term sustainability. I've been in the industry for over 10 years, and I've had the privilege of building strong relationships with our industry and listening to them and seeing what their needs are to these challenges. We all are facing challenges in the American wine industry, both on the growing side, the winemaking side, and even the selling side, the business side of things, through the tasting rooms, etc. One thing, though, on the viticulture side that we're all concerned about, no matter where you are, in most cases, is water scarcity. And also, how can we do more with less? And so in Texas, I established the most comprehensive irrigation and grapevine water use efficiency study. To date, this is our third season. We're generating a lot of good data that we'll be able to comb through and provide our growers with research-based information on not only the best ways to schedule and monitor irrigation and also water in the soil and vines, utilizing a lot of affordable technologies that can help improve our decision making. So we're excited to be able to continue with this work, to be able to get this information in the hands of our growers that they can add to their vineyard management protocols as it relates to irrigation and with a focus on water conservation.
Napa County's picket fire began in late August in the rural hills near Calistoga. Within days, it had grown to nearly 6,800 acres. Thankfully, the fire spread northeast into less densely planted areas, reducing the damages that could have devastated this year's harvest. The conditions could have easily been much worse, as we saw in 2020 when multiple wildfires swept across much of the West Coast. That year, the wine industry suffered an estimated $3.7 billion in losses. Analytical laboratories became overwhelmed by the volume of samples submitted, with some customers waiting up to a month for their results. When harvest arrives, grape growers and winemakers can't wait weeks for their answers. They need immediate information to decide if their crop can be harvested. Hi, my name is Brandt Barstow, a PhD candidate in the Agricultural and Environmental Chemistry grad group. Working in the Oberholzer Lab and the Wong Lab, my research is focused on developing new tools to address these challenges that the wildfires bring to the North American wine industry. Traditional methods for detecting smoke taint relies on mass spectrometry. While accurate, these instruments are costly to own and operate, and during wildfire events, testing costs and turnaround times can quickly escalate. My work is focused on developing a faster, more affordable analytical method using fluorescence spectroscopy coupled with chemometric modeling. This approach lowers both consumable costs and analysis time. The Aqualog rapidly captures the spectral fingerprints of juice and wine, reflecting its own unique chemical signature and allowing us to detect signs of smoke exposure. With the right modeling, we can screen for smoke risks in near real time, helping winemakers and growers make informed harvest decisions. I am truly grateful to the American Wine Society Educational Foundation and to the generous scholarship in memory of Thomas King.
Hi there, my name is Nikolas and I'm a PhD student at the University of British Columbia in Vancouver. Here in British Columbia, we've been experiencing more frequent and intense heat waves, and this is extremely concerning for grape growers. For example, in 2021, temperatures in several important wine regions here reached temperatures above 45 degrees Celsius. Furthermore, temperatures above 35 degrees Celsius are very common. There are many different methods being suggested to growers for solving this temperature issue, such as increasing irrigation, use of foliar sprays, and different variety selection. And these are exactly what my PhD research will focus on. I will be conducting experiments in controlled growth chambers to determine suitable varieties typically grown in BC that will perform well under heat stress. But I will also be conducting experiments in BC vineyards, where I will be applying heat waves artificially using small greenhouses built from PVC piping and plastic film. The first vineyard experiments will be focusing on biostimulant sprays used as mitigation techniques for heat waves. I am currently testing 3 different biostimulants that are available commercially, and I'm happy to say that we've been making good progress in the short time that we've been working on the project, as we've already sprayed and then subsequently experienced some minor heat waves this summer in the vineyard. My second vineyard experiment involves applying deficit irrigation to 2 different varieties and seeing how these respond differently to heat waves. We are hoping to get insight into how differing hydraulic behaviors of grape varieties can impact the stress they experience during the combination of heat and water stress. So with these research projects, we're hoping to ease the worries of grape growers when it comes to heat waves by finding practical and effective solutions to dealing with this stress. I would like to thank Castellan Lab, where I'm currently doing my PhD, for all their help with this research and their invaluable advice. I would also like to thank the American Wine Society Educational Foundation for giving me this platform to speak about my research. I strongly believe that with proper knowledge, the North American grape and wine industry can be prepared for a changing climate.